Hello and welcome to this Vibrant Publications summary video. Today, we'll be talking about the study called Evaluation of the Vibrant DNA Microarray for the High-throughput Multiplex Detection of Enteric Pathogens in Clinical Samples. Let's get started. Every year, gastrointestinal infections affect millions worldwide, causing not only acute discomfort, but also long-term health consequences like nutrient malabsorption, post-infectious IBS, and even autoimmune complications. Yet, identifying the exact culprit quickly and accurately has always been a challenge. That's where the gut microbiology revolution comes in. Today, we're looking at a major step forward: Vibrant study of their Gut Pathogens Panel, a component of their GutZoomer stool test. This peer-reviewed study, published in Gut Pathogens in 2019, set out to develop and evaluate a high-throughput multiplex DNA microarray test capable of detecting 27 enteric pathogens, including 16 bacteria, 5 viruses, 4 parasites, and 2 fungi, from a single stool sample. Over 1,000 clinical and archive specimens were tested between 2015 and 2017. The method combined DNA and RNA extraction with multiplex PCR amplification and microarray hybridization to produce results with high specificity and sensitivity. More on this in a second. Additionally, in the laboratory validation, this method produced results in hours instead of days, showing how powerful the technology is compared to older culture-based methods. In clinical practice, Vibrant integrates this same high-accuracy platform into the comprehensive GutZoomer test, which requires 7 different methodologies and delivers results within about a week. Real-time polymerase chain reaction, commonly called RT-PCR, is the gold standard for accurate identification of organisms. Briefly, DNA is extracted and purified from stool samples. Then, microbe genes are paired with known complementary sequences called primers to identify which microbes are present. Primers are small fragments of single-stranded DNA or RNA designed to bind specifically to a single region in the microbe genome, allowing for precise amplification of the diagnostic target area. Then, probes are used to quantify the amount of each microbe. Probes bind to the target area and give off a fluorescent signal as amplification of that area increases. This fluorescent signal is then read by the quantitative real-time PCR machine where the reaction is occurring to get a quantitative readout. The results of the study were striking. Vibrant's methodology had a sensitivity of 95.9% and a specificity of 100%. A sensitivity of 95.9% means there is a 4.1% chance of a false negative. In other words, there is a small chance you may have a pathogen even if it wasn't detected by the stool test. A specificity of 100% means there are 0 false positives. In other words, if the test shows you have a pathogen, you 100% have a pathogen. What's more is that the limit of detection was as low as 10 to the second cells per milliliter for some bacteria and parasites. In other words, Vibrant can detect pathogens even at very low levels, which means you don't have to be super sick with tons of overgrowth to get a positive test result. The most frequently detected pathogens were Candida and E. coli species, including E. coli 0157 and EPAC, with 20% of positive cases showing polymicrobial infections, meaning patients had more than one pathogen at the same time. Importantly, the assay also detected hard-to-capture organisms like Helicobacter pylori and Listeria species. These are species not included in many competing panels. Many of the pathogens highlighted in the study are part of Vibrant's GutZoomer, an advanced stool test that expands beyond infection detection to assess the overall microbiome, gut health markers, and functional imbalances. Using the same high-accuracy microarray and RT-PCR methodology, the Vibrant Wellness GutZoomer measures over 200 microbes, plus a broad array of gut health markers, such as gut inflammatory markers like calprotectin, digestion markers like pancreatic elastase 1, immune balance markers like secretory IgA, gut antibodies like fecal antigliadin, malabsorption markers like vegetable fiber and fecal fat, gut metabolites like bile acids and short-chain fatty acids, and gut diversity indices. This thorough assessment offers an unparalleled view of gut function and its impact on overall health. This means clinicians can move from identifying a problem to personalizing solutions, whether it's antimicrobial therapy, probiotics, dietary changes, or other interventions. This study is more than a technical paper. It's proof that high-throughput multiplex pathogen detection is here, and it's ready to change patient care. By accurately identifying more pathogens faster and with greater sensitivity than many existing tools, Vibrant's Gut Pathogens Panel, and by extension, the complete Gut Zoomer, helps clinicians act quickly and effectively. If you're a healthcare provider, consider microbiome testing for patients with unresolved gastrointestinal symptoms, or as part of preventative healthcare. The full published study and the Gut Zoomer sample report are linked in the video description, so you can see how research-grade science translates into everyday clinical action. To learn more, visit the links in the video description. If you have questions, please ask them in the comments below this video. Thanks for watching, and I hope you have a vibrant day.